Welcome to PGOG Pathways to Growth in Obstetrics and Gynecology for Postgraduates and Consultants. Today's topic is Physiology of Menstruation or Regulation of the Menstrual Cycle. Now, comparatively, it is such a basic topic, and one might think that it will be very easy to approach the topic, but there is explosion of newer information especially related to the regulation of menstrual cycle. There are so many molecular changes which have been identified in the last few years that when we start to prepare or try to understand the topic, we find sometimes we might find it a complex one. And that is the reason with this thought that I am approaching this topic so that to make it as easy as possible for you. Now, the ovarian cycle, we can divide it into follicular phase, ovulation, and luteal phase. That is how we divide it. And in our undergraduate years, especially, we know that there is a follicular phase, then there is ovulation, then there is a luteal phase. But postgraduate level, remember that we need to identify another part of the ovarian cycle where there are important events which are taking place as far as the regulation of the menstrual cycle goes. And that fourth component is luteal follicular transition. So when as a undergraduate student you say that ovarian cycle has three components: follicular phase followed by ovulation, and then the luteal phase and menstruation occurs, and then again there will be a follicular phase, only three. But as far as for the ovarian cycle postgraduate level, remember that the fourth one which needs to be added is luteal follicular transition. Follicular phase, we can divide it further in greater details as primordial follicle development, where a cohort of the primordial follicles cohort starts developing. Second step is a rescue from atresia. The cohort is rescued from atresia, which is labeled as recruitment of the follicles. Then there is development of or the further growth of the follicles as pre-antral follicles, antral follicles, and antral follicles. From the antral follicles, there is a selection of dominant follicle, and atresia of the rest of the follicles is important step. And once there is a selection, then the follicle further develops into the pre-ovulatory follicle. So these are the important steps to be remembered whenever we want to describe the uh, regulation of the menstrual cycle, because these all these events in the follicular phase are very important, and these there are minute changes which are ultimately responsible for regulation of the menstrual cycle. Important concepts related to follicular phase are primordial follicles start developing as a cohort throughout the life of a woman till menopause. It's not only throughout the life, even intrauterine life from 20 weeks of pregnancy till the newborn baby's delivery. Also, there is a continuous development of primordial follicle with associated with atresia, and that is why the number keeps on decreasing right from intrauterine life. And this process of development of the primordial follicles as a cohort is taking place throughout the life of a woman. Primordial follicle does not need FSH for initial development is another most important concept. I want all of you to understand that follicular stimulating hormone or hypothalamo pituitary ovarian axis normal functioning is not necessary for this initial development of primordial follicle. The initial development is dependent on molecular changes taking place in the ovarian tissue itself by autocrine paracrine system. Now, this is the most important terminology as far as understanding this topic goes. That once we understand what is autocrine system, what is paracrine system, and what is endocrine system, almost it is almost like we are understanding the core of this topic or the main concept related to the topic. So let us see what is this. What is autocrine is a cell is there. It is secreting a substance, and this substance is acting on the same cell via the receptors which are present on the cell wall. This is called as autocrine way of functioning or autocrine system, where the cell itself is regulating its own function with the presence of receptors on the cell wall for the substance, whatever substance is being secreted by that cell. On the other hand, paracrine is whatever substance is being secreted, it is acting on the adjacent cells, cells which are present in the vicinity. So action is on the cells which are in vicinity or surrounding cells, then it is called as a paracrine system of functioning. And whenever we say endocrine, what do you mean by endocrine way of action of the hormones is the cell is secreting this hormone, it enters into the bloodstream and it reaches to the distant tissues and there it is acting on the cells. Then it is endocrine. So autocrine, paracrine and endocrine. And as far as this development of primordial follicle goes, we need to understand that it is not under the influence of endocrine system, it is not under the influence of FSH, but it is under the influence of autocrine, paracrine way of regulation that primordial follicle initial development takes place. Now, what leads to or regulates follicular development? That is a very commonly asked question, and we need to, if we think about it, we always say it is the HPO axis, hypothalamo pituitary ovarian axis, which is regulating the follicular development or which leads to the follicular development. But remember that apart from this HPO axis and follicular stimulating hormone and luteinizing hormone, two important factors which are um, playing a major role in development of the follicle are one is local peptides such as anti-Mullerian hormone, activin, inhibin. These are the local peptides. Now, what do you mean by peptide? Is a small protein. Peptide, the meaning of the word peptide is it is a protein itself, but it is smaller. That means the number of amino acids is less in peptides. Now, these peptides are present in almost all the tissues in the body. Similarly, these are present in ovarian tissue also. And these peptides are regulating the follicular development because of their local action. And the important peptides are AMH, that is anti-Mullerian hormone, activin, and inhibin. So these are autocrine, paracrine regulators which play a major role in or contribute a major role in development of follicle. And last is androgens. Androgens are produced in ovary itself, and they have a major role in the decision whether the follicle will go for atresia or follicle will continue to grow as a antral follicle. So androgens also play important role. So hypothalamo pituitary ovarian system, we know that from ovary there is a positive feedback as well as negative feedback, and that is how the HPO axis is functioning. But local androgens and local peptides also, remember, that play an important role. So if you are asked the question how the regulation of the menstrual cycle takes place, what is mainly uh, governing the regulation of the menstrual cycle, you should not say that only hypothalamo pituitary ovarian axis or pituitary hormones. Remember that you must include the local peptides by autocrine paracrine regulators, which are autocrine paracrine regulators, and you must include androgen which is being secreted in the ovary itself, which also plays an important role. What is a primordial follicle? It is a single oocyte which is arrested in the meiosis surrounded by single layer of granulosa cells. And we say that primordial follicle is the main functional unit of the ovary. Whether follicular growth and atresia stops during pregnancy, if you are asked this question, now as in the previous slide we have already seen that this follicular growth and atresia continues uninterrupted from infancy to menopause. It also continues during ovulation or anovulation. So irrespective of whether the cycle is ovulatory and ovulatory, this follicular initial follicular growth and the cohort which is going for atresia, this process is taking place throughout the life of a woman. So number of primordial follicles at 20 weeks is 6 to 7 million. At birth it is 2 million. So many. If you say around 4 to from 6 to 7 million, it is directly coming to 2 million. That means it is so many 4 to 5 million are going for development and atresia in intrauterine life itself. Then further there is a reduction by puberty to 3 lakhs, and only 400 become dominant follicles. Rest always go for atresia by the time a woman reaches the stage of menopause. How much total time is required for development of a dominant follicle or pre-ovulatory follicle? That means follicle just before the ovulation. Around 84 days is the estimated time required for the development of the follicle from initial um, uh, from onset of development. There is a initial development, then there is a rescue from atresia by the action of follicular stimulating hormone, which we call it as recruitment, and then further development occurs up to pre-ovulatory stage. Here, as you can see, this is the primordial follicle, single layer of granulosa cell which is surrounding the oocyte. Early development takes place up to 70 days. And this early development is not hormone dependent, it is autocrine, paracrine dependent. So early development is of 70 days. That means whenever there is a recruitment takes place of the cycle in early part of the menstrual cycle, already the growth has occurred for previous almost more than 2 months, 70 days. The development is a single follicle which is growing is already in a stage of development for 70 days. Then only it is ready for the recruitment. That means under the influence of follicular stimulating hormone, instead of going for atresia, it goes and further develops into a pre-antral and antral follicle, and one follicle will become a dominant follicle normally in a menstrual cycle. Now, as I have said in the first slide itself, that there are important molecular changes which are regulating the development of the or regulating the menstrual cycle, and even regulating the follicular development. And especially for primordial follicle development, that means early development, there are important molecular changes. It is independent of follicle stimulating hormone. Again and again, I am repeating it because it is the most important concept, and I hope that I will repeat it for more a number of times. I will repeat it, it will become clearer to you that initial 70 days of follicular development is not under the influence of hypothalamo pituitary ovarian axis. It is not dependent on follicular stimulating hormone. It is only the local molecular changes which are governing the initial 70 days, almost 70 days of follicular development. So what are these molecular changes? The most important are transforming growth factor beta proteins. It is a family of protein which is called as super family. Why it is called as super family of proteins is there are so many proteins which are present all over the body in various tissues which come under the umbrella of this TGF beta proteins. That is why it is called as super family of proteins, and it is called as protein or peptide. And as I have already said that at some places in the books you will find it called as peptides. So peptides are nothing but proteins which are smaller in amino acid number. So these super family of proteins are produced locally and they act as autocrine paracrine system. There are certain TGB beta proteins which are enhancing the primordial follicular development, and certain proteins which are inhibiting the primordial follicular development. Enhancing are activin. Those which are enhancing the primordial follicular development are activin and bone morphogenetic protein, BMP, whereas inhibiting are inhibin and AMH, anti-Mullerian hormone. So this is important to remember that two are enhancing, two are inhibiting. Name is easy to remember, inhibiting, inhibin, and anti-Mullerian hormone. Again, something anti is there, that means it will also be inhibiting. And opposite is activin. It enhances the follicular development, primordial follicular development, and bone morphogenetic protein. These are the four important for initial development of primordial follicle. Apart from this TGF beta proteins, there are certain neurotrophins also which are needed for primordial follicle development, for the primordial follicle to be to develop and get converted into primary follicle. For this, what is required is this TGF beta family proteins and neurotrophins. All these are locally produced and regulated in the ovary. These are not secreted anywhere else, but these are acting as autocrine paracrine system. Now, two main molecular changes are responsible for this primordial follicle development. First is we have already seen role of TGF beta family proteins and neurotrophils, and second is there are gap junctions are developing between the granulosa cells and oocyte itself. Now these are the granulosa cells which are present at the periphery, and in the center there is oocyte, and in between this granulosa cell and the oocyte, gap junctions develop with the development of proteins. Gap junction also develop with a development of certain protein, and this gap junction through the gap junction the secretions are going from to and fro from oocyte to the granulosa cell. So there is a bilateral passage of the molecules, and these gap junctions are regulating the growth of each other. That means oocyte is regulating the growth of granulosa cell. Granulosa cells are also responsible for the development of and growth of the oocyte. So there is a bilateral relationship between these two. It is also another example of paracrine way of mechanism, that these are the two cells. One cell is oocyte, other cell is granulosa cell, and they are regulating each other with the help of these gap junctions, and that is why it is an example of paracrine way of regulation of the cell metabolism and cell development. First change which occurs in primordial follicle development is granular cell shape changes. The shape of the granular cell changes from spindle cell to cuboidal. Spindle shape is like this, circular with narrowing at the both the ends is spindle shape. It changes to cuboidal shape of the granulosa cell. So first is initially is not a direct proliferation of granulosa cell. First changes, there is a change in the shape of the cell from spindle shape, it changes to cuboidal shape. Now, how the primordial follicle changes to primary follicle is there is a granulosa cell proliferation. From first changes, we have already seen spindle shape will go for cuboidal. This is like spindle, and now this is like a cuboidal. I hope you understand or appreciate the difference, that cuboid spindle is narrowing at the end, whereas cuboidal is like a cube. So the first initial changes, granulosa cells are becoming cuboidal. Then followed by that, there is a granulosa cell proliferation. Blue ones are granulosa cells which are proliferating. Initially, there is a pseudo stratification, and gradually there is a 3 to 6 layer of the granular cells are forming with the further proliferation, and the cells from the surrounding stroma are differentiating into the theca cells and forming a theca interna and theca externa layer. And together, when the whole structure develops like this, then we call it as a primary follicle. And the size of the primary follicle is almost double of the primordial follicle. Primordial follicle is around 50 micrometer, whereas this uh, primary follicle is a double, that is around 100 micrometer. This change is independent of FSH or HPO axis. That is, again, I have given a star because it is the important concept. Now, what is follicular recruitment? A cohort of primordial follicles start developing. A cohort is a group. Cohort of 3 to 11 follicles per ovary. In both the ovaries, a group of primordial follicles starts developing. This cohort reaches a stage where, unless rescued by FSH, apoptosis occurs or atresia occurs. The literal meaning of apoptosis is beautiful. It is like falling off. That means how the leaves are falling off from the trees. So it is almost like it is natural death, and there is a difference between this apoptosis and cell death by necrosis, that this is physiological cell death. Apoptosis is physiological cell death, and this almost all the primordial follicles which start growing, they are all destined actually for atresia or apoptosis. Actually, the whatever cohort starts developing, almost in 90% of the cases, what will happen is these will go for atresia. Only a few will be rescued, and this rescue of the cohort of follicles at luteal follicular transition. This rescue is possible only at luteal follicular transition, and this rescue of the cohort is known as recruitment of follicles. Now, otherwise, what we feel is recruitment is as if there is a group of or a cohort of follicles, and the follicles grow further, then that is why what we call it as rescue. But the main difference is the concept is it is being getting rescued by going to rescued from what? Rescued from apoptosis. Rather than going for atresia, rather than going for apoptosis, this group of follicles are being subjected to the FSH rise in the uh, luteal follicular transition time of the cycle, that is almost end of the one menstrual cycle and initiation of the second menstrual cycle, that is called as luteal follicular transition. Here the FSH rise is correct, and if the follicle cohort reaches to that stage of development because of the primary follicle development, because of the paracrine autocrine system, and then gets subjected to the FSH, then only there is a further development, that is called as recruitment. So what do you mean by recruitment of follicles? It is a group cohort of the follicles which are natural tendencies to go for atresia, but this atresia is being prevented because the cohort is being subjected to a follicular stimulating hormone rise. There is an increase in FSH in the body that leads to prevention of atresia. Size of the follicle at the time of rescue is 2 to 5 millimeter. Rise and persistence of raised FSH prevents atresia. There is a rise and there is a plateau also is required. Some persistence of FSH for certain time is required for this atresia to be prevented and recruitment to take place. So this is to make it further simpler for you. This is the menstrual cycle. Menstruation has taken place. This is the time of ovulation. Again, menstruation, ovulation, and menstruation. Now there are cohorts which are going for the growth every few days. Almost every few days, the cohorts do start developing. But what happens is whenever a cohort is not subjected to FSH rise in the luteal follicular transition, it will go for atresia. So all these cohorts will go for atresia. Only the cohort which is being subjected to FSH rise in the luteal follicular transition will go for the further development. Then we call that there is a recruitment of the follicle. I hope it makes it easier for you to understand that throughout the life the follicles are developing, primordial follicles. Majority are going for atresia, only few are going for recruitment, or few are getting rescued by FSH to go for atresia, and then they go for further development. Again, the first change is from spindle cell to cuboidal cell. First 70 days growth is by local proteins, that is autocrine paracrine system, primordial follicle in cohort. This is the primordial follicle in cohort. And then when there is a FSH rise, then it goes for recruitment. It is easier to understand when we have the pictorial look in front of our eyes, and that is why I'm drawing this simple diagrams for you, so that if you read as a theory, it seems somewhat complex, but when we visualize it like that in front of our eyes, then the things become simpler to understand, and that is why I am using these line diagrams. What decides the number of follicles being recruited in a cycle? It is the number of inactive follicles available together in both ovaries. How many follicles are available, inactive follicles, that decides how many follicles are being recruited in a cycle. For example, number reduces after unilateral oophorectomy. So primordial follicle cohort, how many follicles will be there in the cohort, that itself depends on how many are the inactive follicles remaining in together in both ovaries. That decides. If one ovary there is a oophorectomy, unilateral oophorectomy is done, it leads to the reduction in the number of follicles which are present in the cohort is the concept. Which follicle in the cohort becomes dominant? Follicle which is ready by autocrine paracrine microenvironment, when stimulated, it becomes dominant. Now, the next stage after the primary follicle, we have already seen diagrammatically also the difference between primordial follicle and primary follicle, and further follicular growth takes place, and then it leads to a stage which is called as pre-antral follicle. This is a stage after follicular recruitment. Here there is a granulosa cell proliferation. Now it is dependent on FSH. So endocrine system is now coming into the action, that for the development of pre-antral follicle, follicular stimulating hormone is necessarily to be secreted by pituitary. There is a synthesis of estrogen, androgen as well as progestin by the granular cell, but mainly it is producing the hormone which is being produced at this stage is estrogen. The most important is estradiol, which is being secreted by the granular cells under the influence of follicle stimulating hormone. Estrogen enhances cell response to FSH. So as the estrogen secretes, it acts on itself, the same cell, so that it increases the number of FSH receptors, and it makes the granular cell, more sensitive to the FSH. So even if the follicular stimulating hormone level is low, still it will respond to the FSH because of this increased sensitivity by the presence of estrogen. It is an example of autocrine mechanism. Now, by this time, zona pellucida surrounds the oocyte, so that the size of the oocyte here onwards remains stationary after the preantral follicle. There are FSH receptors which are present on the granular cells, and this FSH, this estrogen is the one which is mainly making the FSH receptor number increase and makes the granular cell more sensitive to the FSH. So its main role of estrogen is it enhances the cell response to the FSH. Autocrine synergistic action is there for the action of FSH on a granular cell, and follicular cells respond to FSH by paracrine action in absence of FSH receptors. Now, so many granular cells are there. All the granular cells are not necessarily having FSH receptors. There are some granular cells which are not having FSH receptors, but through the paracrine action, the signal goes. Whatever is the action of the FSH on this cell, same action goes through by paracrine action to the adjacent cell, and that is how there is a synchronous growth of all the granular cell. Granular cell proliferation, granular cell maturation is synchronous because of this paracrine action. So this is a beautiful example of autocrine mechanism of estrogen on itself to increase the FSH sensitivity, and paracrine mechanism by which the granular cells are synchronously increasing and proliferating because of this signal which goes from one cell to the other by paracrine mechanism. Even in absence of FSH receptor, this cell will also will react in the same manner as the cell with FSH receptor to follicular stimulating hormone. Now, the fate of preantral follicle, now at every stage, remember that these almost like these all these primer cohort, this primordial follicle which is growing at each stage, always the natural inclination to is to go for atresia, and it needs a specific mechanism for it to stop the atresia. So initial primordial follicle stage, we have already seen that unless FSH is they are getting exposed to FSH, they will all go for atresia. Now here preantral follicle, the important hormone which decides whether the follicle will go for atresia or will continue to develop is androgen. The hormone which decides is androgen. There are around 1500 follicle stimulating hormone receptors per granulosa cell in the this preantral follicle. There are so many receptors develop, FSH receptors, rich in FSH receptor, these granulosa cells. Androgen receptors are also present on granular cells. FSH is needed for aromatization of androgen to estrogen in granular cell. What FSH does is it converts androgen to estrogen with the aromatization. Aromatase enzyme, the androgen is getting converted to estrogen, and for that what is needed is FSH. There is a high FSH and low LH environment is necessary for this aromatization. LH should be low, FSH should be high. Now, whenever low androgen is there, but the low level of androgen is present, then the androgen is getting converted to estrogen. But if the androgen production increases, androgen production in the granulosa cell is high, then instead of going for aromatization and estrogen formation, it gets converted to 5-alpha androgen by the action of the enzyme which is called as 5-alpha reductase. Now, this 5-alpha androgen, it does not it prevents aromatization, one thing, and second is it does not go for further growth, it goes for atresia. So that is why whenever the androgen content is high, the pre-antral follicle will go for atresia. Whenever androgen content is low, further growth of the pre-antral follicle to the antral follicle will take place. So low androgen, estrogen conversion; high androgen, 5-alpha androgen, no estrogen conversion. 5-alpha androgen is not getting converted to estrogen, and it prevents aromatization of this low, whatever androgen is getting aromatized, that also is getting inhibited by 5-alpha androgen. So if it is androgen dominant microenvironment, follicular atresia takes place. Here, what is happening is an androgen dominant microenvironment. Whereas whenever it is a estrogen dominant microenvironment, further growth of the follicle takes place. Follicular growth continues. So this is important concept that from androgen dependent microenvironment, pre-antral follicle must get converted to estrogen dominant microenvironment for it to have a continued development. Otherwise, it will go for atresia. Next is the antral follicle. Here, the extracellular cellular fluid starts getting secreted in the granulosa cells, and gradually the fluid accumulates and it forms a cavity which is called as antrum. Follicular fluid forms antrum. It divides the granulosa cell in the cells which are surrounding the oocyte, which is called as cumulus, oophorus, and rest of the granulosa cells. Estrogen, growth factors, FSH are present in follicular fluid. Follicular fluid is rich in all these hormones as well as growth factors. LH is absent in the follicular fluid till mid-cycle. If premature LH or androgens are present in follicular fluid, it will go for atresia. There has to be FSH and estrogen dominance for further follicular growth. Now, this growth of antral follicle is dependent on a system which is called as two-cell, two-gonadotrophin system. We will see what it is. So remember, for initial primordial follicle development, it is FSH independent. It is dependent on paracrine, autocrine system. Then a stage comes when FSH is necessary for recruitment of the cohort. After that, pre-antral follicle development is dependent on androgens. Now we are coming to antral follicle, where the further growth is dependent on a system which is called as two-cell, two-gonadotrophin system. What are the two cells? Theca cell and granulosa cell. And what are the two gonadotrophins? LH and FSH. So what is happening? Granulosa cells have only exclusively FSH receptors. Only FSH receptors. Whereas theca cells have only LH receptors. So whenever LH acts on the ovary, it will act only on theca cells. Whenever FSH is coming from anterior pituitary and through by the bloodstream, it reaches the ovary, it will act only on granulosa cells. Under LH, theca cells produce androgens from cholesterol. From cholesterol, the theca cells are producing androgen. These androgens enter into the granulosa cells, and under the influence of FSH, granulosa cells convert this androgen to estrogen. Two types of estrogen, estradiol and estrone. These two estrogens are being synthesized in the granulosa cells under the influence of FSH because of the conversion of androgen to estrogen by aromatization. LH plays an important role in final stage of antral follicular development. So whatever estrogen microenvironment is there, it is dependent on this production of androgen by LH. So for the final stage of antral follicular development, this rich source of androgens is necessary. And this source of androgen is because of the action of LH, and that is why LH plays an important role in final stage of antral follicular development. Next is selection of dominant follicle. It is based on two actions of the estrogen. One is the action of the estrogen in follicle itself, and other is the action of the estrogen on pituitary. In the follicle itself, what is happening is follicle having maximum granulosa cell proliferation has maximum estrogen production. Local autocrine paracrine molecular action increases estrogen content. So it is the granulosa cell proliferation which is producing maximum estrogen, as well as local autocrine paracrine molecular action also is contributing to the estrogen content of the follicle. High estrogen increases FSH receptors and increase sensitivity to FSH. The follicle which will have high estrogen will have maximum sensitivity to FSH. Follicle having maximum FSH receptors and intrafollicular estrogen becomes dominant follicle. So it is almost like all the primordial follicles are going for primary follicle, then these are going for pre-antral follicle, then antral follicle, and it is almost like a race. All the antral follicles are competing with each other, and the follicle which has maximum number of FSH receptors, which has maximum granulosa cell proliferation, which has maximum estrogen content, that follicle becomes dominant follicle. Now, second action of estrogen is its action on pituitary. With this increasing secretion of estradiol from these granulosa cells, there is a negative feedback of this estrogen on pituitary for the secretion of FSH. So there is a fall in FSH level. So what happens is dominant follicle growth continues because it has more FSH receptor, and it has a greater sensitivity, maximum sensitivity to FSH. So in spite of falling FSH level, the growth of dominant follicle is uninterrupted, but rest of the follicles go for atresia with the fall in follicle stimulating hormone. This is pictorially again we will see the same thing, why there is atresia of non-dominant follicle. This is the dominant follicle, and these are the smaller ones are non-dominant. As you can see, granulosa cells are more here, proliferation, estrogen is also more here, FSH receptors are maximum in the dominant follicle, and ultimately there is a what is happening here in these non-dominant follicles is low estrogen content, low FSH receptors, and high tumor necrosis factor, high TNF in the non-dominating follicle. And even anti-mullerian hormone is getting secreted by this dominant follicle, and by paracrine action it acts on the lesser follicles, which accelerates atresia of lesser follicles. So ultimately, increase in tumor necrosis factor, fall in estrogen, fall in FSH receptors, along with the AAMH. All these four are contributing for this atresia of the non-dominant follicle. Even inhibin B is now getting secreted by this dominant follicle, which acts on pituitary. It has a negative feedback. It further leads to reduction in FSH, and this negative feedback of estrogen and inhibin B both lead to profound fall in FSH. And with the fall in FSH, these non-dominant follicle are all going for atresia. Dominant follicle has FSH and estrogen synergy, and this synergy leads to development of LH receptors also. There is LH receptor emergence on theca cells of dominant follicle. So towards the end of this development of the dominant follicle, there is another action of FSH is development of LH receptors on theca cells of the dominant follicle. So how the LH surge takes place is on the basis of this feedback mechanism, HPA axis feedback, we can understand. There is a estradiol has a negative feedback for the FSH on the pituitary. And there is a sustained negative feedback when there is a combination of estradiol and inhibin B. So inhibin B is the type of inhibin or the protein or peptide which is being secreted in the follicular phase of the menstrual cycle by the granulosa cells, and inhibin B has a strong negative feedback action on pituitary. It reduces the level of FSH. So there is a drop in the level of FSH because of the action of this estradiol and inhibin together, inhibin B together. Now, what is happening to the LH by estradiol is when the estradiol level is low, there is a negative feedback of this estradiol for the LH secretion by pituitary. So when low estradiol, it has a negative feedback. LH secretion is reduced by pituitary. But as the estradiol level increases with the development of the dominant follicle, and more estradiol is being produced by the follicles, and whenever estradiol level is greater than 200 picogram per ml, and if that rise in estradiol is remaining for greater than 50 hours, so sustained and profound increase in estradiol is there, then from negative, the feedback changes to positive for the secretion of LH, and that leads to LH initiation of LH surge. And when it happens, this positive feedback for LH, when it happens, the dominant follicle size is around 15 millimeters. Role of peptides and growth factors in regulation of menstrual cycle. We have already seen the role of peptides in the initial development of primordial follicle, where we have already seen 4. Activin and bone morphogenic factor are enhancing the primordial follicle development, whereas inhibin and anti-Mullerian hormone are inhibiting the primordial follicle development. Now, even in the later part of the menstrual, this follicular development also, these molecules or peptides do play a role. So peptides, that is smaller proteins which are present in all the tissues of the body. Similarly, these are present in ovary also. And most important is inhibin. There are 2 types of inhibin, inhibin A and inhibin B. Inhibin B is secreted by granulosa cells under the influence of follicle stimulating hormone. It inhibits FSH secretion from pituitary. So FSH is responsible for its synthesis by granulosa cells, but as the granulosa cells secrete inhibin B, it has a negative feedback on pituitary, and it reduces the FSH secretion by the pituitary gland. GnRH and epidermal growth factor reduce inhibin B secretion, whereas this insulin-like growth factor 1 increases inhibin B secretion. So here there is important growth factor is insulin-like growth factor 1, which is increasing the inhibin B secretion, and as the inhibin B secretion will increase, there will be a fall in FSH. LH releases inhibin A from corpus luteum. So first of the half of the menstrual cycle, remember there is action of inhibin B on pituitary. Second half, there is action of inhibin A because LH is releasing, it helps in synthesis of inhibin A from the corpus luteum, whereas FSH leads to the synthesis of inhibin B by granulosa cells. So blue is the graph for inhibin B, and the purple is for the graph of inhibin A, which is exclusively present in second half of cycle, whereas inhibin B is mainly present in first half of cycle. It is easier to remember pictorially like this. Inhibin A also suppresses gonadotropins from pituitary in late luteal phase. Because of this inhibin A action, there is a fall in the FSH as well as LH, leading to fall in estrogen, progesterone, and that is how the next menstrual cycle starts unless conception takes place. Activin is present in ovary as well as anterior pituitary. It enhances FSH action. Third is follistatin. Follistatin is present in the pituitary as well as in follicular fluid in the ovary, and it suppresses FSH released by the pituitary. Now there are so many growth factors which are mentioned in the textbooks, which are regulating the menstrual cycle, or which have a role to play in the regulation of menstrual cycle. Most important is insulin-like growth factor. And for insulin-like growth factor, there are 2 types, IGF-1, IGF-2. Human ovary mainly has IGF-2, insulin-like growth factor 2. As the name suggests, because it is a growth factor, remember that it will promote the this granulosa cell proliferation. So it promotes granulosa proliferation, aromatase activity, and progesterone synthesis. So what is the action of insulin-like growth factor 2? It promotes granulosa cell proliferation, aromatase activity, that is conversion from androgen to estrogen, and progesterone synthesis. Its receptors are present on theca cells, granulosa cells, as well as luteinized granulosa cells. So these receptors are present in all these three cells, and that is how it has an action in for the all these three mechanisms. Second is epidermal growth factor. Third is transforming growth factor. Then is fibroblast growth factor, platelet derived growth factor, and angiogenic growth factor, which mainly helps in the development of blood vessels inside the follicle. All growth factors are involved in granulosa and theca cell proliferation, hormone production, and molecular local level of follicle growth. So I think it is important to remember that most important is insulin-like growth factor, but you should remember these other factors also. Together, these are all helping in the granulosa and theca cell proliferation, hormone production, as well as molecular level of follicular growth. Next is further development of this antral follicle takes place, and the dominant follicle is now called as the pre-ovulatory follicle. Main changes are granulosa cells they do enlarge in size with lipid inclusions, which is called as luteinized granulosa cell. Theca cells, there is a vacuolation with rich vascularity development, so that this pre-ovulatory follicle comparatively has greater blood vessels as compared to the antral follicle. Oocyte, in the oocyte, there is important changes. Meiosis resumes. There is resumption of meiosis with completion of reduction division. So first polar body develops, and that is how there is a completion of reduction division. Estrogen peak is seen 24 to 36 hours before ovulation. LH surge starts. LH luteinizes granulosa cells with production of progesterone. So before ovulation also, we always know that progesterone is mainly the hormone which is being secreted by corpus luteum. But remember that it is also present, it has a vital role before ovulation. Few days just before ovulation, progesterone also comes into the picture. And how it is getting secreted is, it is getting secreted by these luteinized granulosa cells. Progesterone is in low dose here, but it in low dose it helps in LH surge. Androgen rise with atresia of smaller follicles. Androgen is also more in the pre-ovulatory follicle, which helps in atresia of smaller follicles, and it increases libido in this part, so that the conception rate increase in the conception will be there because of this natural increase in libido. So this is the first polar body, and reduction division gets completed with 23 chromosomes. This is the corpus, this cumulus ophorus. These are the granulosa cells which are getting enlarging with lipid inclusions, which are called as luteinized granulosa cells, which are secreting progesterone in small quantity. Theca cells and granulosa cells. Theca cells are mainly androgen producing. Androgen is coming from theca cells, which helps in the atresia of non-dominant follicles. Is more and more estrogen is getting secreted, and that high level of estrogen for greater duration leads to positive feedback on pituitary with LH surge, and LH is only responsible for this progesterone secretion by luteinization of granulosa cells. So these are the important hormonal changes which are seen in this pre-ovulatory follicle development. Now the important event is ovulation. Timing of ovulation is, it is 34 to 36 hours after onset of LH surge. It is the most important reliable indicator. 24 to 36 hours after peak estradiol level, and 10 to 12 hours after LH peak. Here, as you can see, this is estradiol peak. 24 to 36 hours later, ovulation takes place, whereas after LH peak, usually 10 to 12 hours later, ovulation takes place. This is mainly the important diagram which each one of you should be able to draw also. It is the important one, which is once you are able to draw this diagram, that means you have understood regulation of menstrual cycle. So as what you can see here, the first hormone is, this is the green one is FSH. So, there is a slight increase in the initial part of the menstrual cycle, then there is a fall. Then around it, just before ovulation, there is a peak of FSH, then sustained fall in the second half, and then as the menstruation cycle ends, the FSH starts rising. Red is the LH1, which is low initially, and then there is a sudden LH surge. It falls, but it remains slightly higher in the second half, and then it starts increasing again towards the end of cycle. This blue one is estradiol. Low estradiol initially, then there is a rise, fall, and second peak is seen in the second half of cycle, where the estradiol is being secreted by corpus luteum. Progesterone, this is important. Before ovulation itself, it starts rising by luteinized granulosa cells. It has an important role to play, that it positively leads to LH surge. Unless this luteinization of granulosa cells take place, LH surge will not occur and ovulation won't take place. So progesterone plays important role here, and there is a sustained increase in second half of cycle, and then there is a fall with the corpus luteal atresia, or the demise of corpus luteum. Both this estrogen and progesterone both will come to a almost negligent level, leading to the onset of next menstrual cycle. Now, what is the action of LH for ovulation? There is a main important one is resumption of meiosis takes place, luteinization of granulosa cells we have already seen, synthesis of progesterone, and synthesis of prostaglandins. Prostaglandins are very important for ovulation to take place, and this synthesis of prostaglandin is the action by LH, and that is the prostaglandins play important role in the ovulation, and that is why it is recommended that prostaglandin synthetase inhibitors should not be given in cases of infertility, because it will hamper the this ovulation to take place, because the prostaglandin will not be acting for, it is necessary for ovulation. So if by mistake infertility patient or any patient is taking, woman is taking prostaglandin synthetase inhibitors just at the expected time of ovulation, ovulation will fail to take place, that cycle will become anovulatory. Progesterone action, it helps in digestion and rupture of follicular wall by enhancing action of proteolytic enzymes and prostaglandins. This progesterone is very important for ovulation to take place. Prostaglandin synthetase inhibitors are to be avoided in infertility cases. Then another action of progesterone is it leads to mid-cycle rise in FSH by positive feedback. As you can see here, FSH, which was low, it has again another spike here just before ovulation. It is because of this action of progesterone. So progesterone helps in the ovulation by enhancing the action of proteolytic enzymes and prostaglandins for rupture, this for ovulation to take place. And secondly, the progesterone helps in the mid-cycle rise of FSH. Now, why FSH is necessary? FSH frees oocyte from follicular attachment. From surrounding granulosa cells, the oocyte is freed because of the action of FSH. It converts plasminogen to plasmin, which is necessary for ovulation. It ensures adequate LH receptors for normal luteal phase. So this FSH has important actions. Because of this FSH, there are adequate LH receptors in the corpus luteum, leading to adequate luteal phase with adequate secretion of progesterone and estradiol. Ovulation is not an explosive event. It is a slow process with complex hormonal and molecular changes. Now coming to the luteal phase. Corpus luteum formation is because of the luteinization of granulosa cells after the ovulation. Then differentiation of theca cells from surrounding stromal cells. There is further differentiation of theca cells to form the corpus luteum. There is vascularization with filling of antrum with blood by VEGF. So there is vascularization with filling of this antrum with the blood, and this action is by the vascular endothelial growth factor, which is being secreted by this uh, corpus luteal cells. So now some of the textbook mention this stage of corpus luteal development as development of corpus hemorrhagicum. Avascular granulosa is getting converted into vascular luteal cells by the development of blood vessels. Vascularization takes place. Small blood vessels are there, and ultimately the blood is getting collected in the this antrum also. Corpus luteum has highest blood flow per unit of any tissue in the body. So this is the peculiarity of corpus luteum. Remember that it is so rich in the vascularity and it has a highest blood flow per unit in body. So it is that vascular. Rupture of this corpus luteum leads to acute emergency with development of hemoperitoneum, and this risk increases in women who are on anticoagulant, and this especially occurs after 1 week of ovulation when the vascularization is maximum, and that is why it is an indication for these women who are on anticoagulant to give them hormonal treatment for suppression of ovulation. Otherwise it can lead to life threatening uh, bleeding and hemoperitoneum in a um, menstrual cycle. This simple event itself can lead to life threatening condition because of this anticoagulant treatment. This corpus hemorrhagicum will rupture and it will almost like a picture, acute picture like a ectopic pregnancy. It is a close differential diagnosis of ectopic pregnancy. Optimum pre-ovulatory follicle or optimum presence of FSH action is necessary for normal luteal phase. This is another important concept, that so many times we say that there is luteal phase insufficiency. So it is not the corpus luteal insufficiency, that means corpus luteum is insufficient. It depends on how the follicle grows. How many FSH receptors are there in pre-ovulatory follicle? How much is the granulosa cell proliferation, how much is the FSH production, that is optimum, then only the corpus luteal function is normal. So follicular development decides whether corpus luteum will have a normal function with a production of adequate hormones or inadequate corpus luteal activity. Important facts related to corpus luteum are its lifespan is dependent on small amount of LH which is present in the second half of the ovulatory cycle. Uh, in the say that in the second half there is a LH surge, but there is some amount of LH remains persistently raised, and this small amount decides the lifespan of the corpus luteum. If this small amount of LH is not there, there will be earlier demise of corpus luteum. Lifespan is usually 11 to 17 days, and we say that it is persistent 14 days. We take it as an average. Second half of the cycle is usually persistent. We say that the menstrual cycles are sometimes becoming irregular when there is a change in uh, duration of follicular phase, and usually the luteal phase is remaining persistently of 14 days, and it is given that 11 to 7 days 17 days we can consider this duration as a normal lifespan of the corpus luteum and normal duration of luteal phase. Corpus luteum itself is a complex and heterogeneous structure. It has luteal cells which are producing steroid, that is why these are called as steroidogenic, but these are present only 30% of the corpus luteum is formed by luteal cells, whereas 70% is formed by endothelial cells, leukocytes, and fibroblasts. Endothelial cells contribute for 35% of the total cells present in corpus luteum. In steroidogenic cells, we have two types of cells, small and large cells, which are secreting various different hormones. Small cells are secreting only LH or SCG, whereas large cells are mainly secreting all the other hormones such as estradiol, progesterone, inhibin A, prostaglandin F2 alpha, and vascular endothelial growth factor. All these are mainly secreted by large cells. There is abundance of small cells. It secretes only LH, but there are gap junctions, and that is how these small and large cells together are mainly regulating each other's function also because of the gap junction. And that is why, though the small cells are in abundance and large cells are less in number, still the secretion of all these hormones is taking place normally because of the presence of these gap junctions between the small and large cells. Endothelial cells, leukocytes, and fibroblasts are non-steroidogenic cells. Leukolysis occurs because of the local estrogen, then presence of nitric oxide and prostaglandin F2 alpha. So for nitric oxide, prostaglandin F2 alpha, all these are contributed by these non-steroidogenic cells also. Now, if pregnancy takes place, then SCG after conception prevents luteolysis. This corpus luteal lysis is prevented by the presence of SCG, and that is called as corpus luteal rescue. Next is a luteal follicular transition. This period is from decline of estradiol and progesterone in late luteal phase to the selection of dominant follicle for next cycle. So that means from end of luteal phase and initiation of follicular phase. This is called as luteal follicular transition. And important events what are happening is end of luteal phase there will be decline in estrogen progesterone, and initiation of follicular phases by selection of dominant follicle. So this is a gray zone between the luteal and follicular phase, which is very important for the regulation of menstrual cycle. Here the menstruation starts, then there is a selective increase in FSH. There is no disproportionate increase in FSH as compared to LH. Only FSH increases during this luteal follicular transition, and this increase in the FSH is because of the demise of corpus luteum. That means corpus luteum demise takes place, corpus luteum stops secreting estradiol and progesterone as well as inhibin A, and that leads to the FSH rise. How it leads to FSH rise is when the estradiol and inhibin A secretion is reduced, it leads to removal of negative feedback on pituitary. That is how there is an increase in FSH. Secondly, removal of estrogen progesterone negative feedback on hypothalamus increases the GnRH pulse frequency, increasing the FSH. So why FSH rise is there in luteal follicular transition? It is because of this removal of negative feedback on pituitary as well as hypothalamus. FSH rise leads to recruitment of 70-day-old follicle cohort, and dominant follicle gets selected from this cohort, and next menstrual regulation of next menstrual cycle takes place, as we have already seen in the initial slides. Thank you so much. Mainly I have prepared this topic from Spirop's textbook of Clinical Gynecological Endocrinology and Infertility 2019. Thank you once again. Hope you have understood this complex topic. Any questions, any queries, or your comments are most welcome. Thank you once again.